Hello friends, in this video I would be teaching you all about molecular basis of inheritance. So let's get started with the video. A bacteriophage phi 174 has 5386 nucleotides. Bacteriophage lambda has 48502 base pairs. Escherichia coli has 4.6 into 10 to the power 6 base pairs, and haploid content of human DNA is 3.3 into 10 to the power 9 base pairs. Learn all these facts. Okay. Uracil nitrogenous base is present in RNA. Uracil is present in RNA at the place of thymine, which is present in DNA. A nitrogenous base is linked to the pentose sugar through N-glycosidic linkage to form a nucleoside. Nucleoside is formed by sugar plus base. Okay, nitrogenous base. A nucleotide, a phosphate group is phosphate group is linked to the 5-hydroxy of a nucleotide through phosphoesterase linkage. So, a nucleotide is formed by sugar plus nitrogenous base plus phosphate. Okay, the backbone of polynucleotide chain is formed due to sugar and phosphate, and backbone is due to sugar and phosphate, and a nucleotide contains sugar, nitrogenous base and phosphate. Uracil in RNA. 5-methyl-uracil is another chemical name of thymine. 5-methyl-uracil is the name of thymine. Watson and Crick proposed double helix model for the structure of DNA based on X-ray diffraction data produced by Wilkins and Franklin. Adenine forms 2 hydrogen bond with thymine, and guanine forms 3 hydrogen bond with cytosine. Okay. Learn this fact. The 2 chains of DNA are coiled in right-handed fashion. The pitch of the helix is 3.4 nanometer, and there are roughly 10 base pair in each turn. 10 base pair in each turn, right-handed fashion, and the pitch of the helix is 3.4 nanometers. Francis Crick proposed a central dogma which states that genetic information flows from DNA, RNA to protein. Central dogma says DNA to RNA to protein. Okay. In prokaryotes, DNA being negatively charged is held with some proteins that are positively charged in region termed as nucleoid. In prokaryotes, it's a nucleoid which serves as nucleus. In eukaryotes, DNA is held with set of positively charged basic proteins called histones. They are organized to form a unit of 8 molecules called histone octamer. What they are called? Histone octamers. The DNA is wrapped around histone octamer to form structures called nucleosome. They will ask the composition of nucleosome. It contains 200 base pairs of DNA helix. Okay, like DNA is wrapped like this. Okay, and H1 histone is here. Nucleosome constitute the repeating unit of structure in a nucleus called chromatin. They are seen as beads on a string. What? Beads on a string when viewed under electron microscope. The chromatin fibers coil and condensed at metaphase stage of cell divisions to form chromosome. The packaging of chromatin at higher level requires proteins called non-histone chromosomal protein. What they are called? Non-histone chromosomal proteins. Okay. In nucleus, the region of chromatin that are loosely packed and stand light are referred to as euchromatin, and the chromatin that are densely packed and stand dark are called heterochromatin. Densely packed and stand darks are heterochromatin, and the loosely packed and stand light are called euchromatin. Euchromatin is transcriptionally active and heterochromatin is inactive. One minute. Heterochromatin is inactive and euchromatin is active. Okay. In Griffith's experiment, Streptococcus pneumoniae bacteria were used. S strain of bacteria produces smooth colonies because they have mucopolysaccharide coat that were absent in R strain. So here you can see Streptococcus pneumoniae was used by Griffith, and S strain bacteria produce smooth colonies because they have polysaccharide. In Griffith experiment, mice infected with S strain bacteria die from pneumonia infection. Hersey and Chase worked together to discover the nature of transforming principle. They grew virus on medium containing radioactive phosphorus and some of the medium containing radioactive sulfur. Phosphorus and sulfur were used, which were radioactive. In Hersey-Chase experiment, the sequence of stages were infection, blending, and centrifugation. Infection, blending, and centrifugation. Infection, blending, centrifugation. At the end of radioactive sulfur were detected in supernatant and radioactive phosphorus were detected in cell. 2' OH group present at every nucleotide in RNA is reactive group and marks RNA labile and easily degradable. DNA nucleic acid is preferred for storage of genetic information and RNA nucleic acid for transmission. You have to learn the fact. DNA for storage and RNA for transmission. Okay. Nucleic acid. RNA nucleic acid was the first genetic material. Which was the first genetic material? RNA was the first genetic material. The replication of DNA is semi-conservative as each DNA molecule have one parental and one newly synthesized strand like this. Here you can see like this. So they are semi-conservative in nature. In 1958, Michelson and Schultz have proved that DNA replicates semi-conservatively. Michelson and Schultz proved that DNA replicates semi-conservatively. Michelson and Schultz experiments, the heavy DNA molecule was distinguished from normal DNA by centrifugation in cesium chloride density gradient. Cesium chloride was used by Michelson and Schultz. The main enzyme for replication of DNA is DNA-dependent DNA pol. Replication requires DNA-dependent DNA polymerase. In E. coli, the average rate of polymerization is 2000 base pair per second. The replication of DNA occurs within a small opening of DNA helix referred to as replication fork. The polymerization occurs in one direction, that is 5' to 3'. The DNA replication in the templates with 5' to 3' polarity is discontinuous, and discontinuity synthesized fragments are joined by enzyme DNA ligase. DNA ligase acts as the glue. Okay. The process of copying genetic information from one strand of DNA into RNA is termed as transcription. That is DNA to RNA. Okay. A transcription unit of DNA is defined primarily by three regions, that are promoter, structural gene, and the terminator. Promoter, structural gene, and the terminator. During transcription, the DNA strands having polarity 3' to 5' act as template strand. You can learn it by 3 starts with T and template also start with T. So 3 you can learn it by this T and T 3. Okay. Coding strand of DNA is used as reference point while defining transcription, and its polarity is 5' to 3'. Okay. T for 3, T for template. Promoter is a DNA sequence that provides binding site for RNA polymerase and is located upstream of the structural gene. Like promoter, structural gene, and terminator. Terminator is a DNA sequence that defines the end of the process of transcription and is located downstream of the coding strand. In eukaryotes, the coding strands are called exons and appears in mature RNA. Mature RNA contains exons. They are interrupted by introns. Introns are non-coding regions, non-coding sequences, and exons are coding sequences. Exons are coding sequences and introns are non-coding sequences. Okay. In bacteria, during transcription, RNA polymerase facilitates opening of the helix and continues elongation. Who helps in elongation? That is RNA polymerase. RNA polymerase is associated with transiently with sigma, which is called initiation factor, and with rho, which is called terminator factor. Initiator factor is sigma and the terminator factor is rho. Termination factor rho and initiator factor is sigma. In eukaryotes, RNA polymerase 1 transcribes rRNA, RNA polymerase 3 transcribes tRNA and 5S RNA and snRNA, and RNA polymerase 2 transcribes mRNA. You can learn it by RMT, RNA pol 1, 2 and 3. Okay, RMT is the mnemonic for this. And learn this fact also. Splicing is the process where introns are removed and exons are joined in a definite order. Introns are removed and exons are joined. In capping, an unusual nucleotide methyl guanosyl triphosphate is added at 5' end of hnRNA, and tailing adenylate residues are added at 3' end in template independent manner. Okay. Capping and tailing. George Gamow, a physicist, suggested that in order to code for all the 20 amino acids, the code should be made up of 3 nucleotides. This is the fact given by George Gamow. The codon for amino acid is triplet. The number of codons that code for amino acid is 61 and 3 codons are stop codons. 3 are stop codons and those who codes are 61 amino acids. One codon codes for only one amino acid and hence it is unambiguous and specific. Some amino acids are coded by more than one codon and hence the codon is degenerate. Learn the terms unambiguous, degenerate. AUG has dual functions. It codes for methionine and also act as initiator codon. What AUG does is codes for methionine and act as initiator codon. Insertion or deletion of one or two bases in a gene changes the reading frame. Such mutations are called frame shift insertion or frame shift deletion. Transfer RNA act as adapter molecule. tRNA or soluble RNA act as adapter molecule. Okay, there is no tRNA for stop codons. The secondary structure of tRNA looks like a clover leaf, but in actual structure of the tRNA, it is compact molecule which looks as inverted L-shaped. The process of charging of tRNA is called amino acylation of tRNA. The cellular factor responsible for synthesizing proteins is ribosome. Ribosome is the protein factory. It consists of structural RNA and about 80 different proteins. 80 different proteins. 23 rRNA bacteria is the enzyme called ribozyme. 23S rRNA is the ribozyme. Some additional sequences in mRNA that is not translated are referred to as untranslated region, and they are necessary for smooth going process of translation. For initiation of translations, the ribosome binds to the mRNA at start codon that is recognized only by the initiator tRNA. At the end of the translation, release factor binds to the stop codon, terminating translation and releasing the complete polypeptide from the ribosome. The accessibility of promoter region of prokaryotic DNA is regulated by interaction of proteins with DNA sequences termed as operators. In bacteria, the arrangement of DNA sequences in which polycystonic structural gene is regulated by a common promoter and regulatory genes is called operon. What's an operon? The definition is here. The lac operon consists of one regulatory gene, I gene, where I refers to inhibitor. One regulatory gene, that is the I, and here I refers to as inhibitor. In lac gene, Z gene codes for beta galactosidase and Y gene codes for permease and A genes for transacetylase. What? Z gene for beta galactosidase, Y gene for permease and A gene for transacetylase. Okay. Regulation of lac operon by a repressor is referred to as negative regulation. Regulation of lac, okay. So Z gene transcribes or beta galactosidase, Y gene permease and A gene transacetylase. Human Genome Project was launched in year 1990 and completed in year 2003. The genes that are expressed as RNA are referred to as expressed sequence tags. What they are called? Express sequence tags. Assessing different regions in the DNA sequence with function is called sequence annotation, that you are annotating a particular function to a particular gene. This is called sequence annotation. What it's called? Sequence annotation. In Human Genome Project, the fragments were sequenced using automated DNA sequencers that work on the principle of method developed by Frederick Sanger. Sanger sequencing. In human genome, less than 2% of the genome codes for proteins. The chromosome 1 has most gene, that is 2968, and chromosome Y has the fewest, that is 231. Learn this fact. Chromosome Y has 231 and chromosome 1 has 2968. Less than 2 for proteins. Okay. DNA fingerprinting involves identifying differences in some specific regions in DNA sequences called repetitive DNA. If any inheritable mutation is observed in a population at high frequency, it is referred to as DNA polymorphism. If an inheritable mutation that can be transferred from parents to the child, then the inheritable mutation is observed in a population at a higher frequency, than it's called as DNA polymorphism. The technique of DNA fingerprinting was initially developed by Alec Jeffreys. Learn the name. Alec Jeffreys. In this satellite DNA used as probe, so very high degree of polymorphism refers to as variable number of tandem repeats. Thus, its size varies from 0.1 to 20 kilobase. Okay, so these are the facts from genetics. I hope you liked the session. Codons, triplets. This is all about molecular basis of inheritance. Do like, subscribe to my channel, and thank you and all the best.